Hello, my name is Armaghan Sarvar and I'm a Master of Bioinformatics student at the University of British Columbia and the Bureau Lab at BC Cancer Genome Sciences Center. This is the pre-recorded talk on STASH, a data structure based on stochastic tile hashing, which I've been developing. The decrease in the cost of sequencing over the last decade has led to more accessible generation of reads from DNA and RNA data. Many famous bioinformatics algorithms such as genome assembly or structural variation detection have been based on identifying short, fixed-length substrings of reads, which are called k-mers. Hence, there has been a growing need for developing methods to store these k-mers that can support various queries. For example, as demonstrated in this figure, the genomic reads are segmented into 3 mers, which will be stored to represent the genome. On the other hand, k-mer-based representations are not immune to sequencing errors, and that's why many tools and data structures have been based on using spaces which can tolerate mismatches. For example, the Bloom filter itself is a probabilistic data structure used for fast membership queries that represents data by hashing values as bit array indices. An extension of the Bloom filter is the multi-index Bloom filter, or MIBF, that consists of a vector of identifiers together with the membership bits, and it stores spaced sequences, an example of which has been shown here. The multi-index Bloom filter can be used in applications such as taxonomy assignment. Inspired by these data structures, we introduced a stochastic tile hashing or stashing in order to describe large collections of nucleotide sequences through a lossy representation. The proposed underlying data structure is called STASH, which uses a fixed allocated memory and provides a means to store and query sequence mapping information without any alignment. For example, STASH can answer the question of whether 2 sequences originate from similar genomic regions. But how is the STASH populated? The underlying structure of STASH is a 2-dimensional bit array consisting of sets of T-bit tiles, where the rows or the memory loci are indicated by hashed values of subsequences. Having an input sequence, it is hashed using the NT-hash algorithm with sliding windows of symmetric spaced patterns, and the resulting NT-hash values specify which rows of the memory block to update. Having H spaced patterns, each sliding window position will update H rows of stash, and we call these H accessed rows a stash frame. As for the next step in filling a stash tile, the columns and the stored tile values are indicated by hashes of the sequence ID. So for the input sequence, the sequence ID is hashed twice using 2 different hash functions, and the resulting values are segmented into T1 and T2 bit tiles respectively. The tiles of the first hash function output specify the column, and the tiles of the second hash function output specify the value to be stored. In this slide, the whole data structure and the process of sequence data population have been shown for 4 spaced patterns. In order to combine the NT-hash output values with the 2 sequence ID hashes, we can see that for the ith spaced pattern, the ith most significant bits of the other spaced patterns are concatenated in order to address an index in the sequence ID hash tiles. For example, here, for the first hash function output, the first significant bits of the other 3 hash function outputs are concatenated, which results in 0, 0, 1. So we will address index number 1 in the sequence ID hashes, which leads to filling column 3 with the value 7. In order to answer the question of whether 2 genomic regions are covered by the same set of reads, we define a score metric called the number of matches that captures the similarity between any 2 frames of interest. And it's calculated such that each tile in the first frame is compared to H tiles in the second frame residing in the same column. and it's incremented if there is at least one equal value to the examining tile in the corresponding column of frame 2. Here, I will show an example of how sample stash frames are updated after filling the data structure with a C. elegans Oxford Nanopore long sequencing read. Having the input read, the read ID hashes would be as follows, which will be segmented into 4-bit tiles, and these tiles correspond to these values. Having the sliding window of space seed patterns, the NT hash output values would be as follows, each of which will determine one row in stash. Considering the H, or here 4 most significant bits of the NT hash outputs, we will extract the following bits, which will correspond to these numbers. Having the first hash function output, we will address index number 2 in the read ID hashes and fill column 3 with the value of 15. Having the second hash function output, we will address index number 7 in the read ID hashes, which will lead to filling column 13 with the value of 15. And so on. Considering the second sliding window of space seed patterns, the NT hash output values would be as follows, and we can see that for the first repeated number, we will address index number 2 here and fill column 3 with the value 15. And for the second repeated number, we address index number 6 and fill column 6 with the value 2. So here these two frames have the number of matches of 2, which is because they are both coming from the same read. If values stored in tiles from unrelated read ID hashes are uniformly, independently, and identically distributed, the probability of a pair of tiles sharing the same stored value by chance would be 2 to the power of minus T2, where T2 is the stash tile bits. Then the probability of a tile from the first sequence compared to H corresponding tiles from the second sequence having at least one hit would be 1 minus 1 minus pi to the power of H, which is the match probability, and H is the frame rows. Finally, the probability of two frames having at least m matching tiles would be based on the following formula, which is a binomial distribution, and it is called the Salston score. And Salston score actually implies the relatedness of any two frames. Our expectation for the range of values is that for small distances on a genomic region, the number of matches would be higher because the corresponding frames would be coming from the same reads when filling the stash data structure, and the Salston score values would be lower. On the other hand, for large distances on a region, we expect the number of matches to be lower because there would be no connection between the underlying frames, and the Salston score values to be higher. In order to have a more general view of a genomic region and also ignore sequence errors and gaps, we define windows of frames where each window consists of frames that have a distance of stride with each other. And we compare two windows by counting the maximum number of matches between all pairs of their frames. Stash can get saturated if we keep adding the same k-mers into it. So we define saturation as the probability of a row being filled completely by the space seed outputs. Assuming stash has C columns, the probability of inserting into a row tile would be 1 over C. And if we define the number of insertions of the same space seed as X, the stash row occupancy would be based on the following formula. As we can see in this plot, when coverage increases, the saturation gets closer to 1. And the plot also shows us until which coverage stash can effectively store insertions. If we define the average length of input reads as L average, the number of reads as NR, the number of columns in stash as C, and the number of rows in a stash frame as H, and finally the number of frames in a stash window as NF, the time complexity of filling the data structure would be O of H squared times L times NR, where if we assume H to be a small number, then the time complexity of filling stash is linear with respect to the input data size. The time complexity of comparing two frames is O of h squared times c plus h times k, and the time complexity of comparing two windows is O of nf squared times h squared times c. As for the experimentation, we have filled the stash with the experimental Oxford nanopore long sequencing reads from chromosome 21 of this human individual, and by fixing a window on a randomly chosen position along the reference human genome GRCH38 chromosome 21, and comparing it with a sliding window that moves from the left to the right of this randomly chosen position, we calculate the number of matches and also the Salston score values between them. So here we can see the results. On the left, the number of matches have been shown, and we can see that as the sliding window gets closer to the fixed window, which is on the middle position here, the number of matches values increase, which is because more common reads cover the closer neighboring areas. And on the right, the Salston score values have been shown in logarithm scale. And as we can see, when we get further away from the fixed window, which is again on the middle position, the Salston score values increase, which is as we expected. As for the second experiment, we have filled the stash with the same experimental human chromosome 21 reads as the previous experiment, and by considering these reads, we choose a random position on them, which helps us extract two windows that have a distance of delta with each other. By giving these two windows as input to stash, we can calculate the number of matches between them. And by repeating this whole process several times, we get to a distribution of the number of matches. And on this plot, we can see that how by increasing the delta values, which was the distance between two windows, the number of matches distributions get closer to the one of two unrelated windows that have a distance of infinity with each other. The red lines here show the confidence interval of 80% on the unrelated window distribution, and this whole experiment shows us how stash is able to separate between related and unrelated windows. To conclude, we have introduced a novel hash-based data structure called stash that provides an efficient representation of sequence data with demonstrated utility in identifying regional patterns and overlapping sequence frames without any need for sequencing read alignments. We expect stash to provide a wide variety of benefits in bioinformatics algorithms and genomics research. I would like to thank my colleagues Lauren Coombe, Renee Warren, and Dr. Inanch Birol, and also all the other Bioinformatics Technology Lab members who contributed to this project. My project funding is from NSERC, Genome Canada, Genome British Columbia, and the National Institutes of Health. Our software is available in this GitHub link, which consists of a static library together with a demo code showing its functionality. You can also contact me through this email address if you had any questions. Here, the information about other talks or posters from our lab is available. Thank you very much for your attention.